Hi, my name is Tara Pickens. I work in the College of Agriculture within the Department of Food Science, and my advisor is Dr. Daryl Coburn. My project focuses on characterizing the starch-degrading machinery of the gut microbe Bifidobacterium adolescentis. Here's the gene that we are interested in, the putative APUB gene. And so we see here it encodes for a signal peptide, the amylase catalytic subunit, and 3 CBMs, and the pololinase catalytic subunit. So a little bit about the degradation of resistant starch in the human gut. So it is not degraded at all by the human enzymes. We see here in the top panel, if you were to consume the food, it would remain largely undegraded until it gets to the gastrointestinal tract, specifically the lower intestine, where gut microbes are able to interact with it and degrade it. There are multiple reasons why resistant starch is resistant. This partially has to do with physical inaccessibility and also its complex structure, which consists of both alpha-1,4 linkages and alpha-1,6 branch points. The primary resistant starch degraders, Bifidobacterium adolescentis and our bromii, break down these resistant polysaccharides to the intermediate products, acetate, lactate, and mono and oligosaccharides. Secondary degraders then utilize the intermediate products in a mechanism called cross-feeding, and this yields butyrate, which is positively implicated with human health, and we are interested in how this arises. The methods used here first are codon optimization to ensure that the codons used in Bifidobacterium may not work well in E. coli, and so we want to make sure that we're able to get the expression of the proteins that we want in E. coli. Next, molecular cloning, where we determine where to cut the domains out using the existing NCBI annotation, Cluster Omega to align the domains of interest with homologous proteins that have been characterized, JPRED to predict the secondary structure, and FIRE2 to model the 3D protein structures. Then primer design, PCR confirmation via agarose gels. Next, transformation in using the peptide NHIS vector, and these will be transformed into E. coli 10G cells to produce high copy number of plasmids and E. coli BL21 cells to produce the recombinant proteins. Protein production and purification comes next. The protein production is induced using IPTG. The cells are then lysed after overnight incubation using sonication, and then the protein purification is facilitated by the His tag. A TEV protease recognition site will allow for cleavage of the tag later on. And then for characterization, affinity electrophoresis is used for soluble carbohydrates and pull-down assays for insoluble carbohydrates. Here are the results thus far. On figure 2, on the left side, we're seeing the results from CBM53, and on the right for CBM41A. And so panel A shows the 3D models that were generated using FIRE2. Panel B shows the agarose gels that were used to confirm the presence of the desired PCR products at 237 base pairs and 276 base pairs respectively. These bands were cut out of the gel and purified prior to inserting into the peptide vector and transforming, as PCR optimization did not yield a pure product. Next, C, the LB kanamycin plates were used to select for transformants into 10G and BL21 cells. The SDS page gels were used to confirm protein production and purification via FPLC. The molecular weights of CBM53 and 41 are 10.6 and 11.8 kilodaltons, respectively. The next steps will be to remove the His tag from the proteins using the TEV protease recognition site, and to characterize the binding affinity to the soluble substrates, glycogen, soluble potato starch, amylose, amylopectin, and the insoluble substrates, insoluble potato starch, cornstarch, and high maize cornstarch. Thank you for listening.